Okay, then good morning, everybody. It's a great pleasure for me to welcome you this morning on our session, regulatory requirement. We will try not to make it too much sticking to the title so that you keep awake so early in the morning. I have the pleasure to chair. My name is Clemens Decker-Software. Together with Sean Collins, we will guide you through this session. And first of all, it's a great pleasure to welcome Serge Lyashchenko, who took a long trip from the Memorial Sloan Kettering Cancer Center in New York to join us with this experience with novel radionuclides and radiopharmacy. Please.
Let me just make sure, can the camera see you or?
Yeah.
Maybe it's the screen stuff.
Oh,
Okay, well, but then, do they have a?
Let me see the camera. It's like know how to.
Okay,
On the camera, I see everybody now. It's the whole table.
Okay.
The sound. Yep.
Okay. All right, so good morning, everybody. Thank you for coming here. With us on what's up for dinner yesterday. So as Clemens has mentioned, my name is Serge. I'm a radiopharmacist by training. I work at a cancer center in New York City, which is a pretty big cancer center with a big clinical translation. And since this is a novel radionuclide meeting, and this is a regulatory session, Clemens finally asking me not to speak about technicalities and not too much in regulations. And maybe you could just speak about your experiences, right? And I said, it's a really good idea. I'm going to talk about anything I want. And this is what we're going to do. So we'll talk about experiences in early clinical translation and how the potential relevant regulations, which may be a little bit downstream from what you guys do in terms of radionuclide production, how they may impact how the radionuclide will be developed or possibly not developed, right? So we'll take a look at some examples. In do I?
don't know.
Okay.
I can
Uh,
move for you if you like.
Thanks. So just full disclosure, like I have an academic position, but I'm also involved a little bit in industry. So some of the experiences will be related to industry experiences. Next slide, please. So generally, when we look at factors that are impacting radiopharmaceutical supply, in general, in my experience, it's one of the biggest ones, of course, is the radionuclide availability and accessibility. We'll talk about how that's maybe a little bit different, but nuclear medicine is very, very unique in a sense that in order to have a successful research or even a successful FDA-approved agent, you need access, continuous access to radionuclide. And oftentimes you do not have access. You don't have it, you're going to encounter problems. If you do have it, especially in clinical research, you may actually take advantage of that and do some really cool stuff, as we will see. Of course, as I mentioned, regulations that may eventually impact how the novel radionuclide and the radiopharmaceutical are adopted. We'll discuss it as well. Costs, especially for alphas, especially for novel radionuclide, they're astronomical. So that's also something to talk about. And then in our experience also to make a successful radiopharmaceutical, you need training, you need experience, and you need some common sense sometimes. Next slide, please. Okay, so firstly, let's talk about a little bit availability and accessibility. What do you mean? Availability means like, okay, radionuclide is available somewhere, right? Accessibility means, okay, it's available, but can you actually get it in the lab, right? And the situation is very different from a pharmaceutical company that has a lot of money to a clinical research well-funded institution to a small non-clinical research lab, right? Things are different, right? So depending on who you are and where you're located, you may have some challenges or no challenges with accessibility. Availability usually means due to various factors such as radioactive half-life or whatnot, you just cannot get access to the radionuclide. And what we have in the slide, again, for those living involved in SPECT imaging, my apologies, our clinicians, they prefer PET. So I just put some diagnostic agents or radionuclides in pads. The ones that are in I guess red, you know, somebody somewhere in the US is making them, but you can't really get your hands on it. So for whatever reason, right? So or in a very limited amounts. So they're there, full science. Is it going to go anywhere? I'm not sure. I remain to, again, this is my experience, but I'm not sure if it's going to go anywhere. The stuff that's in green is something that we have already used to either conduct clinical translation, so to get an IND or what's in Europe known as an IND, right? So enabling or we actually use it in the clinic in clinical trials, right? So the stuff in green, you can actually see, you know, what is available pretty much and what we could actually use. This stuff in bold, like the zirconium-89, actinium-225, and bismuth of platinum-12. We could talk about it during this lecture, just because, again, for you guys, it may not be too novel, but for clinical translation work, it is relatively novel, right? Just there's not too many clinical trials exist just yet. Slide. All right, so, and I mentioned regulations may impact the radionuclides developed. So one example, of course, is the actinium-225. We all know various production methods. We all know it's not available and it's really expensive, but you know, one of the DOE-led efforts was like, okay, we'll just take exfoliation material from thorium-232, which has a little bit of actinium-227, and we'll say it's the same as the thorium-229 previous material. And after a while, the FDA actually came back and the regulators said, well, you know, when you convert the radioactivity to small moles of actinium over time, the actinium-227 impurity becomes really, really high. That's true, right? It has a different decay chain. So therefore, you know, you cannot really use spallation material. Well, you can use it, but you have to choose. You either use the pure actinium or you use the spallation material, right? As such, and they came with a bunch of additional requirements, such as one of the biggest ones was actually adaptation on academic centers, because now, in addition to just the CLA review, academic centers, radiopharmacies, they will be obliged to do some ICP mass tests, for example, maybe some alpha spec, most importantly, gamma spectrometry. And the industry is really not great. I mean, for you guys, it's common sense. It's easy. Why don't we use gamma spectrometry? But when you look at traditional radiopharmacy, that was a challenge. And as such, like with the exception of a handful of pharmaceutical companies, nobody actually uses this type of material in clinical trials, right? So they use it in mice. They do preclinical studies, which of course is not the same as using it in humans. But then again, it sort of led to a continuous up until now limitation in supply. Next slide. And it wasn't just academics that were impacted. So this slide is a little bit old, and now when, you know, the last time I presented this slide, somebody from the industry, you know, got up and said this is no longer an issue. Maybe true, but over the last couple of years, you actually have examples, and BMS is not the only example where the Bristol Myers Squibb had to pause the clinical trial just because, because they weren't able to get access to radionuclide, right? So it's really impactful, right? Next slide. All right, so a little bit about Memorial Sloan Kettering. And this is how sort of the continuation of how regulations may impact clinical research, right? So at Memorial Sloan Kettering, we focus mainly on phase 1 clinical translation. So we're not in the business of, you know, developing towards community utilization. We like to push it open to pharma companies to do so. But what's interesting is MSK was actually one of the first centers, and Dr. McDevitt, who's over there on the lower right. They were the first ones to do like actinium and actinium antibodies, right? And all were done for phase 1. Those trials, the molecules were eventually licensed out to pharma companies. So there's some history, but my point is, at the time, this work was done in the fume hood in a regular room. So there was no concept of GMP. They called it like the practice of chemistry. I wasn't even able to find any details, right? So, but now things are changing, right? And the regulators that are demanding more GMP restrictions, more requirements, or flight controls, whatever you want to call them, and they require infrastructure and training to actually bring these agents even for phase 1, right? So what we have been doing over the last 5 years, and I know it seems like a long time, this 5 years, it takes a long time, right? It's MSK decided to actually build a dedicated actinium slash chastity facility to enable manufacture for phase one trials for actinium-225. Now, when you look at this sort of layout, you can see it's not a traditional pharma layout. It's not what in Europe they call full GMP or whatever, right? But it allows us to actually do some phase one manufacturing clinical translation work. Next slide. So these are the sort of the hot cells, I guess you call them, or the production lines that we had one of the radiopharmaceutical manufacturers develop. And remember, I said the process started 5 years ago, right? And we were sort of worried because like it would take so long to build the facility. And at the time when we started, all of the actinium suppliers were like, well, next year or end of this year, we're gonna have, we're gonna find some radium-226, we'll irradiate it in the linac or cyclotron, and we'll have this material. So we're like, oh, like we're way, way behind, right? But in reality, we're just starting to get some actinium. So finally, this was ENM, as an academic institution, we're approached by two entities saying, we have some actinium that's clean. If you wish to have it, you can have it. But before then, over the last 5 years, it's impossible to get, right? You may be able to get a little bit sometimes to do a mouse study if you're lucky. To do a clinical trial, it's exceptionally difficult. So anyway, so that was built. The thing that was interesting is, when we bought the FDG hot cells 10 years ago, the amount of regulatory paperwork that will go with building such and testing such a piece of equipment was minimal. Now it's crazy. They're logging in every serial number for every fuse. They're testing everything. So even for the manufacturers of equipment, the regulatory requirements have increased. Next slide, please.
Sorry. Back.
One more. Yeah.
That one.
Yeah, so this is how the facility looks like. Again, took 5 years to build. It's finally functional, and actually last week we started using one of the chambers, one of the excavators. Like ironically, it was built for actinium-225, but the first radium that we put in there was some very small amounts of thorium-228 and some lead. So which you can see on the lower left. Next slide. So next one. So that's actinium. The story is now we're able to get actinium. Preclinical research is ongoing all the time. Sometime next year, if we have enough interest, we will start making some actinium compounds. But let's talk about lead a little bit. And again, availability plays a key role, right? So if we look 2 years ago, we went to everybody who said, can we get some actinium? They said no, right? So then we went to the OE and we said, can we get some lead-212? They said, sure, but you could also get some thorium-228. So, oh, that's great, because that probably makes a lot of sense. If we figure out a way to isolate the lead from the thorium, that's a win, right? And these days, like I will not go into pricing, but what I could tell you is probably if you were to do like 1 or 2 patients a week, let's say, and you have to choose an alpha, lead-212 would be probably at least 50 times less in costs than actinium-225, right? So there's a huge, huge cost advantage if you're able to actually, and it's a generator, right? 2-year half-life. So if you have more patients, that cost advantage will multiply, right? So it's pretty crazy. So we said, okay, let's think on some lead, right? So next slide. And when we look at the sort of the lead regulations today and the practice of lead, most companies, with some exceptions, they make a certain generator. It could be emanation generator. It could be a radium-224 generator. And they're saying, okay, we're going to make a lot of these generators. We're going to send them to radiopharmacies, and they're going to make radiopharmaceuticals. It's good enough for clinical trials, but then the question then becomes scalability and regulations again, right? Because it's so far, and again, it's going to be corrected, but so far the feedback from the US FDA has been for radiotherapeutics, we will not allow a simple whole kit, right? So the situation may be different in Europe, maybe different outside of Europe, in the US. But again, in the US, they have like, well, it's too much risk. We need these to be essentially manufactured, right? So there's, again, will these light generators go beyond clinical trials? Difficult to say because the regulator may say, well, no, right? So then you have to build like a network of production facilities, which may or may not be challenging. So next slide. All right, so what we decided to do, since we figure, okay, we have somewhat limited budget, we want to have access to lead. Well, let's just get a bunch of thorium from the DOE, and we're gonna figure out a way to isolate the lead, so we can do both non-clinical research and patient doses. And what we sort of came up with is, it's something that's not completely novel, but maybe a little bit novel, where we basically take a thorium 228 in solution, and we apply a bunch of resins to extract out the lead, right? And we do it multiple times to make sure that the thorium 228 is very, very low. At the moment, it's pretty scalable, right? Like so, and a few companies right now, like for example, Orion, they have not the same process, but some similar process. And to me, that is probably the most commercially friendly process, right? So if you look, for example, at gas generators, the issue there is you spot thorium 228 onto some sort of solid matrix. Eventually, the thorium eats into the material and the yields go down, right? So they're limited by the amount of activity you could actually produce. Of course, whatever you produce is very pure, but it's just not enough, right? With this process, you're actually able to make quite a bit, right? And this is, of course, I have to give credit where credit is due. So as I discussed with some of you yesterday, I worked closely with Dr. David Bauer, who used to be in Dresden, I believe, and now he's in University of Missouri, and he did some time with us at MSK as well. So next slide, please. Okay, so moving on from lead and actinium to something that's like a diagnostic type agent, and I'm a little bit biased because this is what the work we have done, but I wanted to sort of give you an example how, there's a perception that zirconium-89 is only good for antibodies and it's limited limitation, but maybe in reality it could potentially have a much bigger use. So several things. I mean, it's no surprise to you to make a radionuclide, right? You irradiate it somewhere, you extract it, maybe somewhere else, right? Then you send it to radiopharmacy and they send it to the plant, right? So Professor Kathy Cutler from the UE, she gave a really good example several years ago. She said, okay, so, we had a company that approached DOE and they wanted to do a holmium-166 drug, right? And holmium-166, I believe, has about a 28-hour half-life, right? So, they tried to follow the model where DOE would send them the radionuclide and then they would make the radiopharmaceutical, send it to the clinic. But they very quickly found out that practically it would be impossible because the drug would be, or the radionuclide would decay, right? So what they have to do is they have to actually build a radiopharmaceutical manufacturing facility and a place where they extract, right? So it's much quicker. So there's no loss during shipping time. And through discussions with various DOE people and, just the community, basically, some people say that, 36-hour half-life is really the minimum that's needed to allow to irradiate a facility, send it to the radiochemical processing facility, send it to the radiopharmacy, and then send it to the end user, right? So concurrently, two things happened. I was involved with Clemens and Renata at IEA, and it became very clear that in developing nations, oftentimes the cost of having a generator or having the radionuclide really prevents to do any PET imaging. So most of those guys, they still do technetium and, PET this both because cameras, don't exist, but also because there's no cheap PET radionuclide available. For 64 is expensive. 18 unit cycle, everything is expensive, right? Generators are expensive. So, and then concurrently, Dr. Mark Bartholomew from University of Hamburg, he said, well, we're taking zirconium oxalate, we're changing it to zirconium chloride, and we're labeling some DOTA PSMA 617, and we're injecting it in patients. And what we have found is that, in early biochemical recurrence patients, where the PSA levels are rising, so a patient already has cancer, you give them a gallium PSMA scan, or you give them a copper, or not copper, sorry, or a 18 PSMA scan, and you see a negative results. Like, in other words, we know patients have cancer, but the diagnostic tracer is not picking it up. Now we're injecting it with zirconium, and then we allow the tracer to biodistribute it, and we image 48 hours later, and voila, the tumor, right? So there's a clinical advantage to having a longer lived radionuclide. So there's a practical advantage, and then there's the potential clinical advantage, right? And now we sort of a little bit see also the copper PSMA sort of saying, well, it may be an advantage because you may be able to image it at later time points, maybe not even in the same day. So all of these things, zirconium needs to have sufficient radioactive half-life, needs to be cheap, right? So when we look at the the potential diagnostic radionuclides, really the only ones that I'm aware of, maybe I may be wrong, maybe there are more. There's the I-124, which has multiple gammas. It has the 600 4 kV gamma. Not that easy to like clinically in a clinical scanner. And there's the zirconium-89. Of course, the disadvantage is, it has the 909 gamma. So people say, okay, there's no symmetry, but it's a cancer patient, so you use risk assessment. So we said, okay, let's just work a little bit on zirconium-89. Next slide. Okay, so and then we did some work, and some of this work was actually already done. So what people have known for a while is the reason why you need to use DFO for antibodies, right? Is because the DFO is a very specific chelator which allows transpilation of zirconium out of the oxidant atom. So when you have zirconium oxalates, when you mix it with like DOTA or DTPA, they don't have the ability to save the zirconium, right? So what DFO does, it's not in a stable complex, but it actually transchelates the zirconium out of the chelate, right? So some efforts have been done to actually isolate the zirconium in chloride form without the use of oxalate, right? So if you avoid the oxalate, you don't get ionization, and hopefully it will label, right? So people have tried it, and it didn't really work. Nobody knew why. And in our zirconium oxalate collection, we actually noticed that there's a lot of iron. We don't know where it comes from, but like everybody saw, there's quite a bit of iron in this presence in this isolated material. Of course, when it's in the oxalate form in DFO, doesn't matter, right? But when you have something that could also bind iron, it's a huge problem. So what we simply have done is just introduce another package which removes the iron. Then we do the same package to bypass oxalate, and voila, we have zirconium chloride, which pretty much behaves as any regular metal would, acetate phosphorus pH 4.5 to 5.5. It labels. So we start labeling first from PSMA molecules, of course, because we now want to study some things, for example, like does the mass of PSMA actually affect biodistribution? Does long-term PSMA INT distribute the same way as 617? And you may say, why is that important? Well, because if you look at clinical trials with lutetium, 617 is doing really well, INT is struggling, and that's because there's some concerns that maybe humans, like the PSMA INT, there's more kidney outtake, so you have to limit the dose, you have to extend the frequency, and this sort of changes things for a clinical trial, right? And that's maybe why that's failing, but maybe truly it's something more like PSMA INT just works, nobody knows, right? There are no investment trials. So anyway, so the lessons learned from there, the radiochemical form and stability are very important, right? After I worked, the several pharma companies reached out and they said, well, we found somebody that's going to be, we read the article, they're making it for it, but it's still not reacting. Why is that? Well, because there may be a stability issue. I mean, zirconium may be hydrolyzed even in hydrochloric acid, and they're just not getting sufficient yields, right? And then the usual suspects rating nuclear purity, specific activity, trace metal, all of that is, of course, very important. Next slide. Okay, so then we said, okay, let's, I mentioned you get a radionuclide and you do some cool stuff to it, right? Like we start developing. So as you guys know, for those of you that are involved in antibodies, usually defaults they want to go to, and then if you want to label antibodies with lanthanides, you kind of have to use either DTPA or you use DOTA or you use some profiles and people use it depends. But anyway, the question then becomes like, okay, the DFO is not exactly the same as DTPA, do they behave different and stuff like that. So what we have figured out is we said, okay, like since we don't need the DFO anymore, right? Because we don't have the oxalate anymore, maybe we could couple it to DTPA and see what happens, which we did. Next slide. And these are the images. So this is impressed, but basically this is a comparison of zirconium DFO, pertuzumab, which you can see on top. We compare it to zirconium DTPA, pertuzumab, which is on the bottom, and we correlated both to terbium-161 biodistribution of DTPA pertuzumab. What we saw is because DFO is a lipophilic molecule, right? So you get higher molecular weight species formation, and you get more liver uptake, which you can see there. With DTPA, of course, it's less. And DTPA was at least on it's just one day, so I'll take it as a absolute truth. But it did predict the biodistribution of terbium-161 better, which is expected because it's an antibody, giant molecule coupled to DTPA. Whatever the metably attached, unlike peptides from molecules, it doesn't really matter that much. Another really cool thing that we noticed is it's actually stable in ascorbic formulation, right? Which is the same formulation used for lutetium and terbium-161. And the specific activities were very high. So we put, I think, about 4 gigas on like microdose levels of antibody, right? Which is giant. Why we did it? Don't ask me. But the nice thing about it is that may enable us to actually label zirconium-89, like on a high specific activity level, and then spike the lutetium, for example, a terbium antibody with the zirconium, so we could actually image, image and see where the antibody goes, because again, the expectation is it's going to go to the same place as the lutetium. Okay, the one thing I sort of forgot to mention, for PSMA zirconium, the difference between the homework study and our method is we're able to put about 10, 15 gigas on like 1 milligram of PSMA, right? And the formulation is stable for about 8 days, like maybe even more. So that allows us to actually make the PSMA centrally and send it to Australia, Japan, anywhere. And it's very, very cheap. So just the pitch is, sorry, that's it. Okay, very shortly, oh my goodness, almost done. They told me to speak for a little bit longer, so should bear with me, probably. But in-house productions facilitates humans, facilitates early production in humans. I think Clemens is going to speak to this a little bit more in the situation in Europe. But what we see, basically what in-house production is, a medical doctor may just order the drug for a patient that's terminally ill as per prescription, and the patient is treated with experimental drug. Really nice. In the US, that's not the case in this life. In the US, they basically says, well, if you need to do a clinical trial, if you need to use it in a human being, you still need an IMPD equivalent, which is an IND equivalent, right? Because they really want you to focus on getting the appropriate indication to make sure that the clinical data supports what you're trying to use the drug for. In reality, what's happening, this was discussed several weeks ago at the SMMI 30 conference, is most pharma companies are going to outside of the US for these early investigations. So they go to Germany, they go to China, they go to India, they go to Australia, and as such, US patients are losing out a little bit, but again, these sort of shows again how regulations impact the development, right? And now there's even talk to make stuff more efficient, so maybe just for imaging agents, maybe in life-threatening diseases, there may be eventually a request to actually change regulations, but for now, it's still a very bureaucratic process. Next one. All right, so a couple of thoughts about operator safety considerations. I think globally this is an issue. I'll tell you a little bit of my experience. Remember I mentioned Mike McDevitt in early actinium trials. When I came from IBM Molecular, which was like a site veteran and cheap producer, I had no idea what actinium 225 was. But I saw like I came from GMP environments, and then they were making actinium in that fume, so I got really scared. I was like, okay, the FDA is going to come in, they're going to shut us down for sure, so I'm going to go clean up, right? So one weekend, I put on gloves, I went in, I cleaned everything up, I found a bunch of tubing and some pipette tips and everything else from the drugs, right? And then a couple of days later, Mike McDevitt shows up and he's like, you dropped my reagents for my actinium labeling. I'm like, Mike, sorry, we'll buy you new ones. So I did. But then fast forward, 8 years, I pricked myself with Ivy, right? And then I started thinking, okay, like, did I get exposed to actinium? And I went to my dad and he said, hey, like, do you think I was exposed to actinium 225 when I was cleaning out? And he said, Serge, let me tell you two things. Number one, I'm very meticulous. I always double bag everything. So unless you open those bags, you should be okay. Secondly, if you were to be exposed, you would know by now, right? So the point of the story is, right? The point of the story is training is absolutely essential because, as these, for you guys are the experts, right? But when you try to penetrate the clinical production of radiopharmacies, it's a different story. Like for us, it's totally wild dollars. Next one. And then, of course, the equipment, alphas especially, we may need to use gamma spectrometry, not just to identify the radionuclide, but to actually run efficiency and quantitate the dose, right? We may need to do fraction collection and gamma counting, and that equipment was never meant for radiopharmaceutical or pharmaceutical production. So when the regulators come in, companies come in, and they say, well, your system is, the electronic system is not pharmacy compliance. And we're like, yeah, we know, but it's like no other choice. So something to consider. Next slide. And lastly, very last two slides, just wanted to mention, a lesson learned, I guess, right? And this, of course, iodine 131 is not a novel compound, but it's sort of an illustration of how we as a community should probably engage the regulators a little bit from the beginning. So these are actually slides from my last PRISMAP lecture, which was online. And for those of you that may remember, it was all halogens. Now it's completely shifted to metals. But anyway, so we had this antibody, right, which goes into brains of patients with neuroblastoma. Next slide. And the clinical data was pretty striking, right? So these patients, it's a terminal disease. So in about 8 weeks or so, that's median survival rate, and all these patients are basically dying. So what MSK decided to do on a compassionate basis, right, is to actually take an antibody which targets the D7H3 antigen, iodinated, radioiodinated, and then inject it into the left meningeal space. And there you could actually see the results, right? So pretty good results. I mean, for a deadly disease, not bad, especially in the 18 months or less, that you could say the survival is over 80%, right? So next slide. And then, so there was a company called YMAPS, and actually the CEO of YMAPS used to be the father of one of the patients that got treated in 2009, and they decided to take this modality to the FDA to get the approval. They actually had to do an additional phase 3 studies, multi-center trial, and some was done in Europe. But the issue was there was no control arm, right? Because this is a deadly disease. The physicians at MSK said, like, we're not gonna just not treat patients. We're gonna try this. And for comparison, YMS decided to use a German registry data, which basically logged patients that got standard treatment. So they decided to use the German data as a control arm, and then as an intervention arm, they would use the MSK data. Long story short, they said, well, you don't have a proper control arm, so you can't really compare. And number two, on Bertamab, or this antibody administration, was one of many interventions, one of which was radiation therapy, right? So you could see resection when possible, irradiation, irradiation, right? They said we cannot statistically just pinpoint the value of Bertamab, right? So therefore, you have to do additional trials. And this, for now at least, it's going through renaissance, but at the time, that was sort of the end of the story. So the point of the story here is, when you have a radionuclide, you may have a perfect drug, you may have a radionuclide, you may have the expertise, but please, when it comes to clinical trials, talk to the regulators first to make sure you have statistically significant, well-designed clinical trials. Because usually those are the only ones that answer the question for sure. So, next slide. And I think that's